Hello everyone, welcome you back. So we have been completing chromatin, we have seen nucleus, chromatin, all those things. So today we have done, already we have done the basics also. So what we need to do today is about the chromosome. And see, chromosome ultimately, chromatin condense and form chromosome, and for every cell, in each and every cell, chromosome is present during the time of cell division. We will be able to see the structure called chromosome. Now to understand chromosome, it's very much important. So structure of chromosome, today we are going to see that particularly about structure of chromosome. Structure of chromosome. Okay, so we will see structure of chromosome now. Fine. See, chromosome, so already you people know very well about chromosome, that chromatin, which is DNA plus histone, is called chromatin. So once again I'll be writing it here because see, first of all it is chromatin. What is chromatin? Chromatin is nothing but DNA plus histone protein. You know that histone is positively charged protein and DNA is negative. So what histone will do? Histone will coil the DNA. So for coiling of DNA, what is required is histone. So histone plus DNA is called chromatin. One more thing I would like to tell you. It's, for example, in case of exam, if they ask you what is chromatin and if they give you options, they will also give you RNA and they will also give you some non-histone proteins also. Now the options, if these are the options, then you need to see that the option wherein all of these are correct. Why? Because you know that DNA's function, the main function of DNA is replication, transcription, all those things. So DNA produces RNA, you people know it. So then chromatin is nothing but DNA. So all those RNAs are also present. Sometimes it might be present there. And some non-histone proteins also will be there. So when we consider the coiling of DNA, we give more importance to DNA and histone protein. But usually if you see chromatin fiber, it can have some non-histone protein. So this is extra. So just remember that it also has some, we can say that it also has some non-histone proteins, and it may also have what? It may also have some RNA material here. Okay, so this is about chromatin, but we give more importance to DNA and histone, which is required for coiling of DNA. Now what will happen? This chromatin fiber, when will you able to see this chromatin fiber? Chromatin you will be able to see it when the cell is in the interphase stage. Interphase stage you people know very well. Interphase stage, what will happen is the cell will be what? It is not actually, they say that interphase stage is a resting stage. However, we will do this in a cell cycle chapter. But still, you need to know this now also. So, interphase stage of cell division or cell cycle is such a stage where they say that it is resting phase. Whereas, it's not resting phase, their cell is not dividing. Okay, cell is not dividing there, but it is not resting also. Metabolically, the cell will be more active in interphase stage. Because in interphase stage only, it will be preparing for the next. And the next phase will be what? Mitosis phase, wherein the cell is actually going to divide. Are you understanding what I am trying to say? Now, so chromatin, when you will see, when the cell is in the stage called interphase stage. Later on, what will happen? Later on, when it enters prophase, which is the first phase of mitosis, during that time, this chromatin will start condensing. Chromatin starts condensing. What will happen to the chromatin? It starts condensing. And when chromatin condenses, what it will form? Chromatin will form chromosome. Chromatin will form chromosome. And that is why we are studying about chromosome today. That is the structure of chromosome, which is very, very important. Now, when you see the structure of chromosome, you people know that chromatin only condenses, right? So chromatin, I'll just show you here like this. Okay, let us consider this is chromatin. Along with what? It's having some histone protein here and there, right? So DNA is coiled around this histone. Let us consider it like this. And now, since we are talking about chromosome here, there will be condensation, there will be more condensation here. Okay, so now we can call it as chromosome. So here what you can see, more coiling of chromatin you can see here. Now we call it as chromosome. Now when this actually the DNA which is present here, it has already replicated in interphase stage. In interphase we have again different stages, G1, S and G2 stages. Okay, there in the S phase the DNA already it has replicated, replication is already done, the copy is already there. Now we need to just condense that also. So when that also get condensed, then what will happen? The same thing, that also get condensed. So here it will show that particular structure also. Along with what? Along with the histone here and there. Histone what? Nucleosomes. We don't know what is nucleosome. 8 histone, that is histone octamer plus DNA is nucleosome. So such type you will find so much of nucleosome here. I am just showing you one, but here you just try to remember that there are totally histone octamers here like this. They are nothing but the histone octamers like this. There are so many histone octamers, you know, that is we call it as nucleosome, right? So we see that nucleosomes are more here and they are completely condensed form, and then we call it as chromosome. In the last class we have done one thing. So in chromosome we have two arms here. This is same like this. This one is same like this. This one is same like this. They both are what? Identical copies. Same thing only. So what do we call it? We call it as chromatids, right? We call it as chromatids here. Both are what? Both are chromatids. Which chromatids? Both are which chromatids? They are sister chromatids, correct? They are sister chromatids. So now, when the chromatin gets condensed, highly condensed, and then it forms the arms called sister chromatids like this, then what will happen? There is a region where it actually gets connected. And this region you can see in the center. So this region is a region where exactly the two chromatids have joined. So briefly I'll be explaining you the structure here, and then we will see in detail. So this region where the two chromatids have joined, this region is called centromere. See, centromere means it is not mandatory that every time it should be in the center. No, it can be anywhere. See, centromere is at last what? It is not particular structure there. It is nothing but it is DNA only. So DNA where it has affinity towards that other sister chromatid wherein it can attach to it. The specific sequences will be there, or specific proteins will be there in that region. That is why it helps in attachment of the other chromatid. So that region can be anywhere on the arms. It can be here, it can be here, it can be there, anywhere. Then what is the function of centromere? The first function of centromere is it attaches the sister chromatids. It helps in attachment of the sister chromatids. This is the first thing. Then the next thing what you need to understand here is that there is a protein-like structure. Okay, so here if you see where there is centromere, okay, here there will be some protein-like structure surrounded to the centromere, and this I hope it is visible to you. It's quite light. Anyways, so this structure, the green structure, what I have done, it is always found attached to the centromere. And this proteinaceous material, it is made up of protein, and we call it as kinetochore. Kinetochore. So what is this kinetochore? Kinetochore is nothing but a protein layer or protein material which is present above the centromere or along with the centromere. Now why it is important? What is the function? What it plays? See, I told you during mitosis, and that too in metaphase stage, chromosomes will come in the equator. Finally, what will be done? During anaphase, the sister chromatids, they will separate from each other. When they are pulled by the spindle fibers, those spindle fibers actually attach to the kinetochore. So they will pull it. Where they are getting attached? They are getting attached to this kinetochore. So then kinetochore is more important here. Fine. So then what will happen? This kinetochore helps in attachment of spindle fibers to it. And finally what it will do? It will pull the centromere along with it and it moves to the opposite pole. This chromatid will move this side. This chromatid will move this side. Because what will happen to this kinetochore? They are microtubules. The spindle fibers will get attached to this kinetochore only, not to the centromere region. Kinetochore is present in the centromere region only, attached to it, but spindle fibers get attached to kinetochore, not to the centromere. Finally, it will help in what? Separating the chromatids. Where exactly at the centromere region it will be separated. I hope this much is clear. So, what we studied now? The central region or any region where two chromatids are joined. This is one thing. That is called centromere. There is one more name for the centromere. Centromere is also called as, primary constriction. It is also called as primary constriction. Now what is this primary constriction? See, what will happen is that I am not able to show it here. Anyways, I'll show it here. See, for example, if this is one chromatid and this is another chromatid, let us consider like this. Both of them have attached here in this region. So what will happen is that this is how you can see there is a constriction here. Because of the attachment, you can see that there is a small groove type structure here. Compression you can see. So this is the primary constriction. This is called primary constriction. And this itself is called what? Because where there is primary constriction, there only you will find that specific DNA sequences on it which helps the chromatid to bind to each other. That is why we call it as what? We call it as centromere region. And one more thing about centromere is that outside the centromere part, what you will see? You will see this disc type structure, which is what? Proteinaceous material. And we call it as what? We call it as kinetochore. I hope this much is clear. So, centromere, just a minute. Centromere, centromere is also called as primary constriction. Centromere helps in attachment of the chromatids together. Centromere helps in division also. That means centromere gets separated easily without any problem. But which structure gets attached to spindle fiber? That is the kinetochore. And kinetochore you will find it always along with the centromere. And now then why it is called primary constriction? Because there is a constriction which has taken place because of that attachment. Then if you consider this name, primary constriction, you can understand one thing. When they are calling this as primary constriction, obviously there must be some other constriction which can be called as secondary, tertiary or something. So that we will see that also. Anyways, this is the basic things which you need to know. So this is centromere, this is kinetochore. These two are what? Sister chromatids. One important thing about this is that these type of structure you will be able to see only during metaphase stage of the cell cycle. Because other than that, you will be seeing chromatin fiber, and prophase it will start condensing. In the metaphase stage, it is completely condensed and they are held at the equator. There if you search and if you find out this chromosome, then you will be able to see all this structure. Now there are some other structure which are important to understand here. So this is done. I hope you people understood this one. Okay, now this chromosome, when it has formed, it will have some outer protein membrane like this. It is having this outer protein membrane like this. Are you understanding this? So this outer membrane It is also made up of protein and we call it as pellicle. What is pellicle? It is nothing but the proteinaceous covering for chromosome. Why it needs to be covered? Because it has genetic material. Nothing should happen to that genetic material. It should be, it should be protected. So there is a protein covering. Then like how you have membrane for other organelles, in the same way, chromosome is also having outer membrane, right? I hope you understood this also. Now, if it is having covering, then obviously the space which is there here, it must be having some material there also. Some enzymes are there because transcription will be happening there, okay? And what you can see, some other things are going on there. Many activities are going on there. Then obviously there will be some kind of proteins, some kind of enzymes, water, minerals, all these things are there. So that is called the ground substance. So just like how nucleus has nucleoplasm, cell has cytoplasm, chromosome, okay, here where this condensed chromatin is here, along with it, it has what? It has this ground substance, and we call this ground substance as what? We call this substance as matrix. Matrix. And what this matrix has? It is the normal matrix, like it has some proteins, it has some enzymes, all these things. It has some, some RNAs, all these things will be there in the matrix. It may be there, may not be there also, but usually it will be there. So this is about the, what you can say, this is about the normal and the general structure. Still there are some more things which we need to understand. First you please take down this much. Okay. Okay, then we will see next. So these are the basic things. Fine. Okay. Next. Okay, what we have done now? Fine. So here what you can see is we have done extra about pellicle, centromere, kinetochore, matrix. Then these structure now, the DNA, it is highly condensed form what you are seeing here. So this is called chromonemata. The highly condensed DNA you are finding here. How you are able to see that? I'll show you here. It's somewhat like this, right? It is like just like a string. It looks like this. And then it is having this beaded structure on it. It has some beaded structure on it like this. Are you able to differentiate? It is looking like a rope, or you can say the string, and some beads are there on it. It looks like bead on string. Beads on string type of structure. So this is nothing but this, right? So now this is called, because it is highly condensed. Once it is put into this pellicle and all those things, we call it as chromatid. Along with ground substance, matrix, along with this centromere, along with this pellicle, completely we call, along with centromere, along with kinetochore, everything, along with everything we call it as what? We call it as one chromatid, the another chromatid. Both are sister chromatids. But only this condensed chromatin, what you are able to see here, it is highly condensed. And now we call this, which is here, which is present here. The same thing is this, okay? The same thing I am showing here. Now this is called what? We call it as chromonemata. What do we call it as? We call it as chromonemata. Singular is chromonemata. How many are there here? Are you able to see two here? In one chromonemata in one chromatid, another chromonemata in another chromatid. So chromonemata, when it is plural, we call it as chromonema. So this is what? This is singular, and plural is what? Plural will be chromonema. No confusions. It's clear. So if you consider this type of chromosome, how many will you have? Chromonemata or will you have chromonema? You will see chromonema. If you are talking about one chromatid, it will have chromonemata. And this beaded structures, what you are able to see here, these beaded structures, we call it as chromomeres. What do we call them? We call them chromomeres. What are chromomeres? Chromomeres are nothing but they are nucleosomes, and we have already done about nucleosomes. Nucleosomes are what? Nucleosomes are 8 histones along with DNA is what? It is nucleosome, right? Do you remember, no? Anyways, once again I'll show you. 4 histones here, the other 4 histones back. Okay, so finally they make what? They make histone octamer, right? And then what is coiled around it? Because it is histone octamer, 8 histones are there. Here more amount of DNA is coiled over here. More amount of DNA is coiled over here. This structure is called nucleosome. And here in this structure you can see many nucleosomes which are together. Many nucleosomes are there because there is highly condensation is done here, no? So more number of nucleosomes you will see together side by side, and those look like what? Beads on a string, and then they name it as what? They name it as chromonemata. I hope this is clear. Now chromosome, where you will be able to see it, all these structures where you will be able to see is in the metaphase stage. Now there is one more thing which is important to understand here. So I'll be rubbing all these things now. So this much I hope it's clear. Now if you see this type of structure, now same thing I'll show you. So this is the normal chromosome, right? This is a primary constriction, and this is the another arm of the chromosome, slightly big covered. Okay, and this region, the primary constriction, this is primary constriction. And what is other name for primary constriction? The other name for primary constriction is what? It is centromere. Okay, this at least is clear. Now for this centromere, there is a protein layer which is surrounding this, the primary constriction. And what is this called as? This is called as primary constriction. I hope you people understood. There is no confusion, right? So this is a primary constriction. This constriction is primary constriction, which is also called as centromere. And this green substance, what you are seeing, it is called kinetochore. What is the speciality of kinetochore? Kinetochore helps in attachment of the spindle fibers to it when the chromatids wants to separate during metaphase, during the early anaphase stage. Then what will happen? So here what you will find inside is, why I am drawing once again is, one thing is because it has become very small and some of you might have not understood it properly, that's why. Okay, so what you will see here, you will see many what? Histone octamers, beaded structures like this. Okay, many you will see in the metaphase stage only. And then, so like this only you will see. So much of these things you will see. And then what is coiled around it? The thing what is coiled around it is what? Is the DNA. I'm just trying to make it more clear, that's it. Like this. And what we call it as? We call it as what? Chromonemata. And when there are 2, we call it as chromonema. Fine. Now, the ends of the chromosomes. Can you see this end? This is one end and this is one end. The end of chromosome, we call it as telomere. What do we call it as? We call it as telomere region. Telomere is nothing but the end of the chromosome. Now, what is the function of telomere? See, what is present inside is DNA. At last it is DNA only. So, DNA will have an end. Okay, and the end of the DNA can attach to the another DNA if there is no telomere. So telomere functions, the end of the chromosome, what you will see here, it is blunt side of the DNA, the blunt side of the chromosome. What it will do is it acts like a cap. It will not allow this chromosome to get attached with another chromosomes. Like, I mean, it's not allowing, for example, if this is a chromosome like this, there is no any other chromosome getting attached like this. No attachment here. Like this, there is no attachment. Why? Because telomere is present here. And telomere region, it acts in such a way that it will not allow the chromosomes to get attached. Or it will not allow the chromosome, it will not allow the chromosome to attach to the nuclear membrane also, because it is present inside the nucleus. However, so during metaphase slowly nucleolus and nucleus will be disintegrating. Anyways, it will not allow that to attach to any other other chromosomes. So this is the function of telomere as far as structure is considered. Okay, and then so many other functions are also there, but for now it's this much is enough. Okay, then what we need to study is there will be in some chromosomes, not all, even in case of humans also, some chromosomes will have this secondary constriction. So this is telomere. What I'll do is I'll just show you this side. This also is telomere only. So how many telomere you will find? Two telomere region you will find. I'll be rubbing this to show secondary constriction. Now some chromosome, I told you, in case of humans also, not all the chromosomes, but some chromosomes will have this secondary constriction as well. Again, like primary only, it will have secondary constriction, and then you will find this bulb-like structure. Okay, now this region is called secondary constriction. Now, is this secondary constriction acts like centromere? No, it will not act like centromere, because kinetochore has affinity only towards the primary constriction region. There are particular proteins which help that kinetochore to attach to centromere region. But here those proteins are not there, so kinetochores will not get attached here, and that is why spindles also will not get attached to secondary constriction region. Okay. And then what is the speciality of secondary constriction? There are some specific DNA sequences present here, which actually secondary constriction, usually they say that secondary constriction region have some kind of DNA, which actually produces this rRNA. And rRNA synthesis, rRNA, what organelle is made up of rRNA? It is the ribosomes. So here, that DNA produces RNA. So this particular DNA which is present when there is secondary constriction, those DNA which actually help in their production of RNA. Which RNA? rRNA. That is why we also call it as NOR region. What do we call it as? We call this area, we call this area as this secondary constriction. Usually it helps in what? This is nucleolus, because rRNA is produced where? In the nucleolus, right? We have already done that. Nucleolus is called as ribosome factory, I told you. Okay, so it is called nucleolus organizing region or organizer region. NOR, what we call it as NOR, N-O-R. Okay, secondary constriction, it may have some other functions also, but usually secondary constriction type 1, there are different types again. So mainly the secondary constriction 1, what you find here after the primary constriction, it is called as NOR region, because it helps in formation of rRNA, or it helps in formation of nucleolus. That is where you will have this rRNA synthesis. That region only will form what? Nucleolus. And there only what will happen? That is the ribosome production, right? So this is called nucleolus organizing region. You will not find the secondary constriction in every chromosome of our cell. Every cell in our body, it has 46 chromosomes. 23 chromosomes from father, 23 from mother. Among those 46, only some chromosomes have this secondary constriction. What you people need to know is this much only. Okay, secondary constriction, We call it as NOR because it helps in rRNA synthesis, which actually functions as nucleolus, which produces ribosomes. Now, after this secondary constriction, what you will find here, this is a bulb-like structure, right? And you can say this is also the end. After this, you will have again telomere. But this bulb-like structure, what you find here, again there are some DNA sequences here. And we call this part as SAT. You know what is SAT? Satellite region. Satellite region. Or you can say, satellite chromosome. Satellite chromosome or satellite region of a chromosome. That means it has a satellite DNA. Now what is this satellite DNA? See, there are some particular sequences here which are repetitive. Okay, it can be short, it can be long. Some, A, T, G, C, right? Adenine, guanine, thymine, cytosine in case of DNA. So those sequences, everywhere those sequences are found. But here in this region, the satellite DNA, here what you will find is, you will find some particular sequences will be completely repetitive for long chain. About, you can say it can be like 2 bases also, like CT, GC, like that, anything. So 2 bases also can be repeated continuously, or many bases also can be repeated continuously. It can be having that 25, 50, 100 repetitions also. That is not for you people. Anyways, see, what you need to remember here is that after the secondary constriction, what region you find in the chromosome, that is called SAT chromosome, or it is called as SAT region of the chromosome, or it contains satellite DNA. What it contains, it is called SAT. That means what it has satellite DNA. Clear? Now what is satellite DNA? It's nothing but just like a normal DNA only, but it has some regions. Some bases are completely repetitive in that particular satellite DNA. And what is the importance? See, satellite DNA, we all have 99%, human beings will have same kind of base sequences. Only 0.1%, 99.9% we all have same, 0.1% it differs. In that, it plays very important role. There are sequences, even we can have same sequences, but the number of repetition will change. Based on that, you can identify different humans. It plays important role in DNA fingerprinting and all those things. Okay, so 99.9% DNA same, there you cannot find any changes. But these are the regions where actually you can differentiate different human beings. So that all things you have in second PUC, don't worry about that. What we need to concentrate here is structure of chromosome. I hope structure of chromosome is clear now. Once again, we will just glance it. It has what? Primary constriction region called centromere. It helps in attachment of chromatids. Okay, and it can be present anywhere on the chromosome. It's not like center means it should be in the center only. No, its main function is it gets attached to kinetochore, and kinetochore gets attached to spindle fibers. And during anaphase, early anaphase, the microtubules get attached to kinetochore and pull them like this. When they pull them like this, the centromere region will then what? Will then separate. Not in case of this. In case of this is the single chromatid I am showing you. It is also chromosome only, but during metaphase stage, this type of structure will be formed already, and centromere will divide, right? Fine. Then we have what? We have secondary constriction. Secondary constriction, it's not present in all the chromosomes. However, it is present only in some chromosomes, and its main function is, it helps in as NOR, that is nucleolus organizing region, where it produces what? The particular sequences of DNA, what is present there, those genes or those things, they code for RNA, and they produce what? They produce rRNA. So we call it as nucleolus organizing region. And then the end of the chromosome, we call it as telomere. It is end. What is the function? It will not allow the chromosomes to get attached. Okay, it will not allow the chromosome to get attached to nucleus membrane or even to other chromosomes. So it helps in protection type thing. The membrane which is present for the chromosome, that membrane is called pellicle. Here we have written. It is called pellicle. And the condensed chromatin which is present in the chromatid, that condensed chromatin, we call it as chromonemata. If two, chromonema. On that chromonemata, you will find this bead-like structures. These are nothing but the nucleosomes. And we call those nucleosomes lot of DNA. I mean, histronoctamer with lot of DNA, nucleosome. So many nucleosomes together, it looks like beads. And then we call it as chromomeres. These only will get stained when we stain the chromosome. And then what we have studied about the satellite chromosome or the satellite region, which has satellite DNA, which has repetitive sequences of particular base or any base pairs. Okay. Then what we said again, kinetochore, which is always attached to where? Primary or secondary? It is always attached to primary constriction, that is nothing but the centromere. And it helps in the splitting of the chromatids. And anything else? Yeah, the ground substance. Like how nucleus has nucleoplasm, cell has cytoplasm, chromosome also have its own matrix material. But remember one thing, what among this chromosome acts as genetic material is only the chromonemata, only that DNA. It will be given, it will be passed on to the next generation, not this pellicle nor the ground substance. Okay, so the main thing is DNA acts as a genetic material here, not other thing. It is just for the sake of division purpose and for the sake of protection purpose, all these things will be there. Finally, what moves on is, we call genetic material as DNA only. Okay, I hope this much is clear now. But we did so much, but we did not discuss who actually termed chromosome, who gave this term chromosome. See, there are scientists who actually discovered it also, but it should not be too complicated to remember so many. There is like Strasburger, Fleming is there, so many scientists are there, but what we need to focus is on what we can remember. Okay, so who gave the term at last? The term chromosome was given by Waldeyer. Check out the spelling. It's Waldeyer. I think it's E. Okay, find out. So Waldeyer, this person, this scientist actually gave the term chromosome. Why did he term it like this, chromosome? See, actually I'm going completely upside down. No, I explained everything and now I'm telling you who actually discovered it or who gave the term. Anyways, see, what we need to remember is, the main thing is this anyways. So why he named it? Because, fine, please take down this much. Okay, so we will see next. See, in NCERT all these things are not given. Then don't think that these things are not important. Okay, these things are important because if you don't know the structure of chromosome, if you don't know all these things, cell cycle chapter, you will not understand anything. So you should know. Okay, so who termed, actually the chromosome was termed by, who coined the term? Coined chromosome. Who was that scientist? His name is Waldeyer. Please remember the name of the scientist. Okay. Now, why did he name it as chromosome only? So why he named it as chromosome? So chroma means what? Chroma. Where you have heard this again? Chromoplast. Colored pigment will be there. In chromoplast, plastids we have done. Chroma means what? It is color. Soma means what? Soma. Soma. Okay. We call autosomes, allosomes. There also we have studied. Okay. Soma means what? It is nothing but the body. He just named it chromosome because when he stained the cell which was about to divide with the astocarmine or astoorsin stains, he saw some stained structures. So he did not know what to name. He just named it as colored bodies. So he named it as chromosomes. So chromosomes means what? They are colored bodies. The colored structures which are present in the cell during that cell division time. Then, okay, they understood all the thing, why it is colored, why it has taken the stain, all these things. Based on that also, again, no, chromosome, I think you understood here. Based on that, based on the staining technique, I mean, based on the staining, Chromatin is again divided into two types. Please remember, this is very, very important. Chromatin, it has two types here. Okay, and the two types are heterochromatin and then euchromatin. Heterochromatin and then euchromatin. What is heterochromatin and what is euchromatin? We will see this, okay?